We came into the project knowing what sequences, what DNA sequences were of interest to us for these different traits that I talked about, disease resistance or ability to grow in harsh conditions. And the process then is first to design genome editing tools, genome editing reagents that allow us to go into a plant genome and make a change at that spot. Okay? We test those reagents and then we move them into a rice transformation setting. Okay. Ultimately, what comes out of that then are little baby plants, and we have to test those plants to determine whether they've got the changes that we wanted to put in there. So the process kind of goes from designing the reagents to target the particular DNA sequence in the genome that you want to change, that you want to edit. Okay. Testing and validating those things work. And then in rice transformation, what's done is we get a sort of a lump of rice tissue to grow from a seed. So you manipulate the hormone concentrations and the media on which you grow this stuff called callus. So you get sort of an undifferentiated mass of tissue. And then we use nature's genetic engineer called agrobacterium to deliver some DNA to these cells in this tissue. And many of the cells in the tissue will take up that DNA. And then we can set the conditions that only the cells that have taken up that DNA will grow. And ultimately, we can regenerate from those single cells that have taken up the DNA new plants. So we go down to a single cell level and then back up to a full plant. Now what happens is when we introduce that DNA itself codes for the genome editing tools. So when that DNA gets expressed, the genome editing tools get expressed within the plant cell. They do their thing. They target the genome sequence that we want to target. And with some frequency, they get the edit that we want, right? Then when we regenerate whole plants from that, we can screen these plants using some molecular screening techniques to ask the question, did this plant get the edit we want? And does that DNA that we introduced there, which encodes for the reagent, the editing tools, does that still persist? Then what happens, we grow that plant up to seed. We self it, that means it crosses to itself and creates new seed. And in that process, random segregation then, in the progeny, the next generation of those plants, we can typically find individuals in which the edit has taken place and the DNA that we originally introduced there to encode the tools is gone. It's just segregated out. And that's what we ultimately want. So we want plants in which the precise edit to the genome is there and nothing else has changed, right? So where are we at right now? We have a table of targets and changes we want to make. And we have designed and built the DNA constructs for the tools to hit each of these targets, and tested each of those in an assay in the lab and shown that they should work just fine. And we've moved about 2/3 of them into that rice tissue culture transformation setting that I was just telling you about. And we have baby plants for about 5 of those now. And we've confirmed the edits in 3. We have to do more screening for the other 2. So it sounds really great right now. But I'll tell you that the first batch through our pipeline has been the easy stuff. The stuff in which the edits are pretty straightforward changes to get out, in which we're essentially removing a word from the book, right? We're not replacing, we're not changing the spelling, we're just removing a word. And that, without getting too technical, is one of the easier edits to achieve. What's really challenging is to take out a word and replace it with a different word, right? Or to change the spelling of a particular word, a particular DNA sequence. That is the sort of the technical frontier right now for genome editing. And we'll see how it goes. I'm hopeful that we have really a great team of people and a lot of expertise in this area in the consortium. So we're hopeful that we'll be able to do this with good efficiency. But that's sort of what's coming up next, right? So we've run through the pipeline for sort of the low-hanging fruit, for the easy stuff, and that sort of sums up our first year. A really important set of next steps is to pursue these more challenging edits, the edits in which we're actually replacing a DNA sequence in one rice variety with sequence from another variety. Okay, so there's sort of what we call allelic replacement, and that's a real challenge. There's some technical hurdles that need to be overcome to make that a more efficient process. So that's an important part of our goals for the next year. At the same time, though, we have material coming through already, in which we've confirmed edits that were easier to make. Just removing a DNA sequence. And so propagating that material, basically growing those plants up for a couple of generations, so that we establish material in which those edits are stable, and then testing that material to determine what we call the phenotypic effects of those edits. In other words, how those edits change or don't change the behavior of the plant. And as I mentioned earlier, we have several different traits that we're interested in running through this genome editing pipeline. So for example, when we focus on our disease resistance traits, the edits that we've made, we would predict would make the plant more resistant to disease. We need to test that out. So we need to do the sort of the characterization of the plants that are coming out of the pipeline. Similarly, we have now plants that have an edit in a gene that confers tolerance to aluminum, right? And aluminum becomes a problem in acidic soils. So we want to ask now whether in this new plant genotypic background, in this new variety, if the change we made gives us the phenotype, the behavior that we want to see, right? And this will all tell us then whether we can actually use genome editing to make specific changes in plant genomes that will result in improvements in plant productivity and plant resilience to stress.